Good evening, everybody. Today I am going to tell about the cell junctions. How we can define a cell junction? So these junctions can also be called as intercellular junctions. I think you know the meaning of intercellular junction. The junction between two different cells is called a intercellular junction. Dep- We can classify these junctions into the tight junction, that is zona occludens. So where these junctions are present, these junctions are present in the apical surface of the cell or the ridges half from the each cell. And these junctions are made up of different proteins such as occludin, claudin, junctional adhesion molecules, etc. The main function of these junctions is it functions as a barrier. Barrier is nothing but it is a gating system which controls and coordinates the different transport of different solutes from in and out of the cell membrane. That we can define as a property of semi-permeability. And the second one, it facilitates the paracellular transport. And a very important function of tight junction is in the central nervous system. If you, it functions as a blood-brain barrier. So this is the structure of a tight junction where we can see a very important protein claudin knots the apical surface of the cell. So this is structure where we can see the structure of the tight junction. Okay, and the empty spaces in between the junction we can call it as a intracellular spaces. We can divide these junctions into cell to cell junction and cell to basal lamina junction. So this the best example for the cell to cell junction is desmosomes, and the cell to basal lamina is hemidesmosomes. So this is structure of desmosome where we can see two cells sitting one after the other, or we can tell as a successive cells, and these cells they are tied together with a tight junction. So such tight knot we can call it as a desmosome, and these desmosomes they play very important role in the transport of solutes, and these desmosomes are made up of a protein called desmoglein and cathedrins. So this is a diagram where we are going to explain the different cell to basal lamina junctions like hemidesmosomes and journal adherens, which is made up of a protein called cathedrin and integrins. So this is the table regarding where the different junctions are present and from which protein it is made up of. The first one desmosome, it is made up of desmoglins which function as a intermediate filaments in the cytoskeleton. Next is the zonula adherens made up of anti-cadherins and hemidesmosomes made up of integrins. It is present in the microfilament. These microfilaments they form the cytoskeleton of the cell. So gap junctions. This is the major junction between the two different cells in a typical human cell, where these gap junctions it plays a very important role in the transport mechanism of solutes from one cell to the another cell. The solute size having not more than 105 nanometers are permitted to transport via cell membrane. So the main aim of the gap junction, the function of the gap junction is these gap junctions they does not allow any type of solute transport having the size more than 105 nanometers.